Hi all. Today's in this video we will start with the small intestine. Okay, so we will look at the secretions of the small intestine, the movements that takes place in the small intestine. Also, we will study about the secretions that comes from the external glands like the pancreas and from the liver into the small intestine. Okay, so let's start first with the secretions of small intestine. Okay, the secretions of small intestines is referred to as succus entericus. Okay, the succus entericus. Okay, so which includes the secretions from the Brunner's gland and the crypts of liver. So when you take like a small intestine, towards the upper side you will see a more of Brunner's gland, whereas when you go to down towards the large intestine you will see a lot of crypts of liver. Okay, so you will study about the histological features of these glands in the histology. Okay, so the main point we have to know is about what are the secretions, the composition of these small intestinal secretions. Of course there are like very high percentage of water. Okay, and then a little bit of mucus is for the lubrication purpose, and it includes electrolytes. Okay, especially you have the sodium, the potassium, calcium, chloride. Okay, so mainly concentrate on the sodium and the potassium. Okay, and as you know, all the secretions include the protective things that is called as the IgA. Okay, the immunoglobulin to protect against the bacteria. Okay, so we consider the small intestine is not like much, there is no much of the bacterial flora, but when we go to the large intestine you will see a lot of bacteria. Okay, the good bacteria that lives in the gut is mainly in the large intestine, and little bit you can also see in the last part of the small intestine in the ileum. Okay, so when we go in the small intestine we have immunoglobulin A to protect against the harmful bacteria. Okay, so these are not like regular things. Okay, so now let us go into the specialized things that is present over here. Okay, so these things like the enterokinase and the digestive enzymes of the small intestine are not secreted. Okay, they are like, for example, this is the cell, okay, of the small intestine. So this is the lumen side and this is the blood side. Okay, so on the luminal side, on the brush border, on the microvilli, on the surface you have these things like enterokinase and the digestive enzymes are the brush border. Okay, so this is referred to as the brush. Okay, the microvilli. Okay, so this is what we call it as the brush border. Okay, so brush border. Okay, so these things like the enterokinase and the digestive enzymes are located on the brush border. Okay, so they are like on the membrane, not getting secreted. Okay, okay, so what does enterokinase do? There is another name for the enterokinase that is called as enteropeptidase. This is the peptide. Okay, enterokinase causes the conversion of trypsinogen to trypsin. We will study later from the pancreas. We have the protein digesting enzyme which is coming as a zymogen. Okay, so the trypsinogen. Okay, trypsinogen, that is the inactive form. The inactive form of the trypsinogen is converted to trypsin. The trypsin is the active form. Okay, the trypsin is the active form, and who caused this conversion? Initially, that is by the enterokinase. Okay, initially by the enterokinase. Later on, once few amount of the trypsinogen is converted to trypsin, what do? Trypsin itself causes the autocatalytic activity. Trypsin itself causes the conversion of trypsinogen to trypsin. Not only that, the trypsin causes the conversion of other protein digesting enzyme into its active form. Other protein enzyme like chymotrypsin, chymotrypsinogen to chymotrypsin, procarboxypeptidase to carboxypeptidase, etc. is done by the trypsin. So trypsin do the remaining. So enterokinase helps to convert trypsinogen to trypsin in the beginning. Okay, so now let's look at the other digestive enzymes. Okay, the other digestive enzyme that is seen in the brush border. Okay, so as I told you, it is not secreted. Okay, but present on the brush border of the enterocytes. Enterocytes means the cells of the GI tract. Okay, of the small intestine we call it as the enterocytes. Okay, okay, and this includes what are these digestive enzymes? Peptidases. Okay, which can cause the conversion of dipeptides or tripeptides into individual amino acids. Not only the peptidases are present on the brush border, you can see the peptidases inside the cytoplasm also. which cause the conversion of the dipeptides and tripeptides. So few amount of dipeptides come in, or when the dipeptides or tripeptides comes on the brush border, it will be converted to individual amino acids. Okay, then the main thing, you have the monosaccharidases, okay, and also lipases. So the lipases breaks down the triglyceride. Okay, so the lipases on the brush border breaks down the triglyceride into the individual fatty acids and the glycerol molecule, and then it is coming in. Okay, the triglyceride is taken in after it has been broken down. Okay, so this lipase is in addition to the pancreatic lipase. You have a pancreatic lipase which is very important, and in addition to pancreatic lipase, you have few amount of lipases on the brush border. Okay, so now let us concentrate mainly on the monosaccharidases. Okay, these are the monosaccharidases, which includes the sucrase, maltase, isomaltase, and lactase. Okay, so this, like for example, sucrase break down sucrose. Okay, sucrose. Okay, so whenever you change the sucrose to sucrase, sucrase is the enzyme ACE. Okay, maltase will break down maltose, isomaltose will be broken down by isomaltase, lactase will break down lactose. Okay, lactose. Okay, so where do we get the lactose? Mainly it is coming from the milk and the milk products. Okay, the lactose. Okay, so what happens in some individuals, as the age increases, okay, as the age increases, normally every person, when the age increases, what happens to the quantity of the lactase enzyme on the brush border? That keeps decreasing. Okay, but in some individuals it will become early. Okay, in the early, like in the adulthood itself, what will happen? The lactase may become deficient. Okay, so they may have like genetically the deficiency of this lactase enzyme. So what happens? They cannot digest the milk. So lactose is normally disaccharide that contains what? The glucose and the galactose. Okay, it contains the glucose and the galactose. Sucrose contains what? The glucose and fructose. Okay, so these are the disaccharide. Maltase and isomaltase is the glucose itself. Okay, two glucoses or the three glucoses together, they are called as the maltase and isomaltase. Okay, so when the person have a deficiency of lactase enzyme, what will happen? The person can get this lactose cannot be broken down. We call that condition as what? Lactose intolerance. Intolerance. Okay, intolerance. Okay, so what is the problem over here? When the person is having a lactose intolerance, the lactose will not be digested in the small intestine. When the lactose is not digested in the small intestine, that goes into the large intestine, and the large intestine contains a lot of bacteria. What does this bacteria will do? Bacteria will start fermenting the lactose. Okay, when the bacteria ferment the lactose, it will produce methane. Okay, and other compounds which is not easy to be eliminated from the intestine, and they are like more gaseous products. Okay, so it will result in bloating. Not only bloating, these lactose molecules, when they are present in the small intestine, they are hygroscopic. They attract water. So these are osmotically active particle, and they will start attracting the water. When they attract the water, what happens? You will have an increased water content in the GI, and the motility of the GI will be increased because of that. So the person can get diarrhea, bloating, okay, the symptoms like that. Okay, so those are the symptoms that is associated with what? The lactose intolerance. Okay, so the best treatment approach is to avoid the lactose containing food. Okay, the lactose containing food is avoided by the patient. Okay, so like milk and the milk products has to be avoided in those scenarios. Okay, important point for your exam also. Okay, the lactose intolerance, you normally get questions on that. Okay, so moving forward. Okay, so what are the functions of these succus entericus? Is to completing the digestion of the proteins, carbohydrates, and the fats. Okay, and how are they regulated? They are regulated by the enteric nervous system. Okay, initiated by the tactile or irritative stimuli coming from the chyme. Okay, so the greater the amount of chyme, the greater the secretions. Okay, so they are locally controlled by the enteric nervous system, which includes the myenteric plexus and the Meissner's plexus. Okay, okay. So now, other than the nervous system, you also have hormones. Okay, to a lesser extent, by secretin and CCK. Secretin is secreted because of the acid from the stomach, Okay, these are happening mainly in the duodenum, and CCK is due to the fat digestive products. Okay, so these will increase the secretion. Whenever you have more acid coming or the fat coming, the secretions, the succus entericus will be increased. Okay, but the secretin and CCK mainly have more function to control the pancreatic secretion and the liver secretion, the bile secretions. Okay, we'll come to that slightly later. Okay, so now let us look into the movements. Okay, so those are the secretions that we have seen. Now let us look into the movements that takes place in the small intestine. Okay, the movements that takes place in the small intestine. Okay, so which includes what? The segmentation contractions and the peristalsis. Okay, peristalsis helps to cause the movement of the food forward, whereas the segmentation contraction helps to distribute the food. Okay, distribute the food. Okay, so in the segmentation contractions, what happens over here? You the tube, for example, you have the bolus over here, where the contraction will take place at the point of the bolus, and on the oral side and the caudal side, what happens on the anal side and the oral side, what will happen? There will be relaxation happening. So what will happen? The bolus will be cut, okay, and it will be converted into smaller particles. Okay, so this helps to distribute, okay, the food particle and helps for the more easy digestion and absorption. Okay, so it consists of alternate contraction and relaxation of adjacent bands of circular smooth muscles, helping in mixing and distribution of food. Okay, regulated by the excitatory and inhibitory neurons in the plexuses. Okay, so these are the myenteric plexus. The myenteric plexus includes the excitatory and inhibitory neurons, which controls the segmentation contraction. Especially the presence of the food itself is the major stimulus for that, and other regulation we will look into. Okay, and next is the peristalsis. Peristalsis is not for the distribution, instead it is for the moving forward of the chyme. Okay, so when you have a chyme over here, where does the contraction happens? On the oral side. Okay, the contraction will happen on the oral side, and then the relaxation happen on the, here you have the relaxation and here you have the contraction. So here you contract and here you relax. So what will happen? The chyme will move forward. Move forward means from the oral side. So for example, this is your oral side and this is your anal side. Okay, so oral to anal. Okay, move towards the anal direction. So contraction happens on the oral side of the chyme, and relaxation happen at the anal side of the chyme. Okay, so the chyme will be moving forward. Okay, so this is the propulsion of the food. So the ring of constrictor waves propelling the food towards the anal direction. Okay, and again regulated by what? The enteric reflexes, the gastroenteric reflexes from the myenteric plexus. Okay, so now in addition to the myenteric plexus, the neuronal, we have what? Hormonal regulation for these movements. Okay, the hormonal regulation. So what are the hormones that will increase the motility? Okay, the increased motility includes gastrin. Okay, so how can you understand this important property of the gastrin? What type of movement is increased by the gastrin? For example, when the food is there in the stomach, okay, so it will cause distension of the stomach and you have more gastrin. Gastrin will come and act on the small intestine and cause the peristaltic movement, cause the movement of the food chyme forward, so that you are expecting new food to come to the small intestine. So in order for the new food to come into the small intestine, you are causing the removal of the already existing food particles in the small intestine. So that is why the gastrin increases the peristalsis, okay, in the small intestine. Okay, now let's look at the CCK. Okay, CCK is secreted in the presence of fat. Okay, CCK is secreted in the presence of fat. Okay, so when the fat is there, you need the lipase to act and the bile to come and emulsify and then cause the separation and increase the chance for the lipase to act. So what does the CCK do? It increases the segmentation contractions. So when it increases the segmentation contraction, it causes the spreading of the food particles. Okay, and helps the increased fat digestion. Okay, so now what does the insulin will do? Okay, insulin is helping to reduce the blood glucose level, right? The role of the insulin is to reduce the blood glucose level. So we don't want like too much of absorption to happen. Instead, we want the peristalsis to happening. So insulin is normally secreted when there is already a high amount of glucose in the blood. Okay, so this high level of glucose will cause what? Increased peristaltic movements within the small intestine. Okay, serotonin is like mostly like a locally secreted over there, okay, and cause like not like a specifically segmentation or the peristaltic contraction. It generally supports the muscular contractions, okay. That's why it increases the motility. So serotonin increases the motility. Okay. Now what are the factors that decreases the motility? Okay, the decreasing motility. Okay, so now you have secretin. So what causes the secretion of secretin? Secretin is secreted by the acid. Okay, the acid is causing the secretion. So acid. So in the presence of acid, we don't want the chyme to move forward. Okay, when the food is coming to the duodenum, and if the chyme contains more acid, we don't want that acid to go further down from the duodenum and cause the damage to the mucosa. Instead, it has to stay there and get mixed with the secretion, the bicarbonate secretions coming from the pancreas and coming from the liver. Okay, the bile contains a lot of bicarbonate and the pancreas contains a lot of bicarbonate. So this bicarbonate has to first neutralize, and once the acid has been neutralized, then what should happen? The food should move further down. Okay, so until then, so whenever you have the secretin, what happens? The motility is restricted, so that it gives the time for the neutralization of the acid. Okay, so now again, what does the glucagon do? Glucagon act opposite to that of insulin. Okay, insulin increases the motility, okay, because you already have a lot of glucose in the blood, but the glucagon is secreted when the blood glucose is very low. So what does the glucagon instruct the intestine? Glucagon tells the intestine to stop the movement, stop the peristalsis, and allow the time to absorb as much as glucose as possible, okay, or as much of nutrient as possible. Okay, so that is why the glucagon is reducing the peristaltic movement. Okay, so the conceptually also important to understand why and how these hormones are causing the regulation, what causes the increased motility and which type of motility is increased. And similarly, when you say the decreased motility, you have to remember what type of motility has been decreased or what may be the cause behind that decrease. Okay, hope you have understood that point. Okay, let's move forward. Okay, so another phenomenon that takes place here is called as what? The migrating motility complex. Migrating motility complex. So this is normally happening in between the meals. Okay, so normally when the meal has been done and you are expecting a new meal to come, then you have the migrating motility complex. Okay, especially for example, before the lunch, almost it is time for the lunch. Maybe you may have the daily habit of taking the lunch at 12 p.m. So what will happen? 12 noon. So what happens at that time? Before, just like few minutes before that, you start to hear some noise and growling sound in the intestine. Okay, so that is due to migrating motility complex. Okay, so the migrating motility complex is a distinct pattern of electrochemical activity observed in the gastrointestinal smooth muscles. Okay, periods between the meals. Okay, it serves to sweep the residual undigested food materials through the digestive tract and hence called the intestinal housekeeping. They just helps to keep the intestine clean. Okay, and migrating motility complex is noisy and are the cause of the sounds. Okay, the growling sounds. The periodic nature of migrating motility complex is controlled by the central nervous system and implemented by the hormone, the enteric hormone. Which hormone? The motilin. Okay, so motilin secreted from the upper portions of the small intestine. Okay, the small intestine secretes the hormone called motilin. Motilin helps to do the migrating motility complex. Okay, so that's about the migrating motility complex. Okay, so next is like this. Now we have finished with the movements and secretions within your small intestine. Now let us see what are the secretions that comes to the small intestine from the pancreas and from the liver. Okay, so next we will go to the pancreas. Okay, so the pancreas includes two parts. Okay, so normally the pancreas have the endocrine part and the exocrine part. The endocrine part is otherwise referred to as what? The islets of Langerhans, which includes the alpha cells, beta cells, delta cells. We have discussed about the pancreas have secreting the insulin, glucagon, somatostatin, etc. So there is the endocrine part. Okay, so endocrine part. But now what we are going to discuss about is the exocrine part. Okay, the exocrine part, which includes the acini. Okay, the acini followed by the duct. Okay, so the acini, the acinar cells over here secretes what? the mainly the enzymes, all the enzymes, okay, and the ducts over here. So this is the acini, okay, and these are the ducts, okay. The duct secretes mainly the bicarbonate, okay. So these are the major components of the pancreatic secretions, the enzymes and the bicarbonate, okay. So the enzymes and the bicarbonates are very important, okay. So in addition to the pancreas, like what are the other things that comes to the duodenum is from the liver, okay. So from the liver you have the right and the left hepatic duct, and you have the common hepatic duct, and the common hepatic duct joins with the cystic duct that comes from the gallbladder. From the gallbladder you have the cystic duct. So right hepatic duct, left hepatic duct, common hepatic duct join with cystic duct will join to common bile duct. The common bile duct and the pancreatic duct coming together and opens into the duodenum, and this opening is guarded by what? A sphincter of Oddi. So the sphincter is regulated. We will study about how this sphincter is regulated, what are the various things that can regulate the sphincter, okay. And this is the pancreas. We have the over the liver secretions and the secretion from the gallbladder or from the pancreas, all of them comes to where? Comes to the duodenum. Okay. So let's discuss about the first about the pancreatic secretions, then we'll go into the liver, okay. So the pancreatic secretions, okay. So we have the two things, that is the bicarbonate and the enzyme. So first let us see the digestive enzyme. So you have digestive enzymes for the protein digestion, for the carbohydrate digestion, and for the lipid digestion. Okay. So for the protein digestion, the major things includes what? Trypsin, that is secreted as trypsinogen, then converted to trypsin by the enterokinase. Okay, and then you have chymotrypsinogen, which is converted to chymotrypsin. Okay, chymotrypsinogen converted to chymotrypsin by what? Trypsin. And then you have procarboxypeptidase A, B, etc. The procarboxypeptidase is converted to carboxypeptidase by the trypsin. Okay, by the trypsin. Good. Okay, so those are the carbohydrate digesting enzyme. Can it cause the complete carbohydrate the protein digestion, and can it cause the complete protein digestion? No. Trypsin, chymotrypsin, carboxypeptidase have like very specificity. In the next slide we will discuss about the specificity. Okay, so now let's look at the other one, the carbohydrate digesting enzyme. What is that? The amylase. Okay, so remember, the amylase can break only the alpha 1,4 linkages and cannot break the 1,6. Okay, the 1,6 cannot be broken down, it will break only 1,4. Okay, and also with the restriction. So that means, can we cause the complete digestion of the carbohydrate like starch, whatever the food that we take? Can we cause the complete digestion? Even the salivary amylase or the pancreatic amylase, both cannot cause the complete digestion. So it will be converted, the polysaccharide will be converted into what? Disaccharide or trisaccharide, etc. So which is then converted into individual monosaccharides by what? By the disaccharidases that you see in the brush border, like sucrase, maltase, isomaltase, lactase, etc. further cause the breakdown. Okay, so that's about the carbohydrate digesting enzyme, the amylase. Lipid digesting is the major one. The number one is the pancreatic lipase. Okay, almost like in a normal person, 80 to 90% of the lipid digestion is done by this pancreatic lipase. Of course, you have the lipase from the mouth or the stomach or from the brush border, but the major one is the pancreatic lipase. Okay, and to break down the cholesterol ester, the bond of the cholesterol with another fatty acid, we have what? Cholesterol esterase. And to break down the phospholipids, okay, we have the phospholipase. Okay, the phospholipase. So these are the major lipid digesting enzymes. Okay, so these are the major digesting enzymes from the pancreas. Okay, also we don't want the trypsin to cause damage, because trypsin is like a protein digesting enzyme. So there are proteins in the pancreas itself. So we don't want on the membrane of the pancreas, etc. you have the proteins, right? So we don't want to damage that. That's why it is being secreted as what? Trypsinogen, not as trypsin. So in addition to that, like as an additional protective mechanism, but we have trypsin inhibitors. Okay, so that prevent the digestion of pancreas itself. Okay, so there is like one specific one called as the alpha 1 antitrypsin. Okay, alpha 1 antitrypsin. Okay, so what does the alpha 1 antitrypsin do? It prevents the trypsin from damaging, so it blocks the trypsin activity. Okay, so mainly what happens is that not only just in the pancreas is this problem. You have a problem when you have a deficiency of alpha 1 antitrypsin. You can get a damage to the pancreas, you can get a damage to the liver, you can get a damage in the lung, or maybe in other tissues also, because trypsin is not something that is coming from only the pancreas. Okay, so for example, in the lung, the neutrophil will secrete the trypsin to damage the bacteria and other proteins over there. But unfortunately, if there is no antitrypsin, or if there is a deficiency of antitrypsin, what will happen? The trypsin, they will cause the damage to the our own tissue itself. Okay, for example, in the case of a lung, you have elastic fibers that helps for the recoiling of the lung, and that is required for the expiration process. So if there is no antitrypsin, the trypsin will cause the elastic fibers to be damaged, or the elastase will cause the damage to the elastic fibers. Then what will happen? The lung can expand, but the lung cannot recoil, lung cannot do the expiration process easily. So what will happen? We will have an expanded lung, something like that is referred to as a barrel chest. Okay, or the condition is referred to as a emphysema. So alpha 1 antitrypsin deficiency can cause emphysema, a lung problem, or can cause damage to the pancreas, or can cause damage to the liver. Okay, so that's the importance of antitrypsin. Okay, but in the pancreas you have a specific trypsin inhibitor. So in case if the trypsinogen is converted to trypsin, another issue is that trypsin is what shows autocatalytic. Not only autocatalytic, it can cause the conversion of all other protein digesting enzyme into its active form. So it is highly dangerous and can cause damage to the pancreas itself and to the other nearby tissues. Okay, so we have discussed about the digestive enzyme. Another major thing that comes from the pancreas is what? The bicarbonate. Okay, so the pancreatic juice is alkaline. Okay, and the pH is 8. Okay, and very alkaline, the most alkaline secretions in our body. Okay, so this is the most alkaline. Okay, and what is the purpose of this? You can see over here, the bicarbonate is very high compared to that of the plasma bicarbonate. Plasma bicarbonate is only 24 milliequivalents per liter, whereas the pancreatic juice have 113 milliequivalents per liter. So it's very high. What is the purpose of this high bicarbonate? It is to neutralize the acid that is coming from the stomach. Not only that, the bicarbonate, when it is being secreted into the ducts and to the lumen of the intestine, what is going to the blood? The hydrogen ion. And what happens to the blood when it passes down through the stomach? It was alkalinized, right? Hope you remember that point. So we call that phenomenon as what? The postprandial alkaline types. The blood that is coming from the stomach will be alkaline in nature. Now what happens when the H plus is going back to the blood? So it will come back to the normal pH. Okay, the blood will also come back to the normal pH because of the pancreatic secretion. Okay, and the quantity is about 1500 ml of pancreatic juice is secreted per day. Okay, so now let us look at the pancreatic enzyme. Okay, and what they do. Okay, the various pancreatic enzyme. Okay, so this table is important. Okay, so we have already mentioned, can this trypsin, chymotrypsin, etc. cause the complete digestion? No, they have specific bond. Okay, you need not memorize like which are the specific bond of each one of them, but you must understand that they don't cause the complete digestion of the pancreas. For example, trypsin will cleave only the peptide bond on the carboxyl side of basic amino acid. Okay, whereas the chymotrypsin will break down on the aromatic amino acid, the peptide bond. Okay, and elastase breaks it the aliphatic amino acid. Okay, carboxypeptidase cleave the amino acid that have aromatic or branched aliphatic side chain. That means all of them, also the carboxypeptidase B, okay, have basic amino acid. Okay, basic side chain. So that means they are not like causing a complete digestion of the protein. So the long polypeptide will be broken down into dipeptides, tripeptides, etc. And the peptidases on the brush border and the peptidases inside the enterocytes, inside the cells, will cause the complete digestion and convert it into individual amino acid. Okay, so you need the other peptidases on the brush border also. Okay, so you have another substance called as the colipase. Okay, the colipase is essential for the activity of the lipase, the pancreatic lipase. Okay, so what happens over here? The pancreatic lipase is coming, and you have the bile that is covering the lipid molecule. So that in order to expose the active site of the lipase, you need the co-lipase. So only in the presence of co-lipase it can cause effective digestion. Okay, so co-lipase. Okay, and the pancreatic lipase that converts the triglycerides into monoglyceride and fatty acids. Okay, monoglyceride, the two monoglyceride and free fatty acids. Okay, so the cholesterol ester, okay, breaks down the cholesterol into cholesterol and the fatty acid. Pancreatic amylase, okay, and the activity requires chloride ions, okay, and breaks down the starch, same as salivary amylase, only break down alpha 1,4, okay, it will break down only alpha 1,4 linkages. Okay, then you have the ribonucleases and the deoxyribonucleases to break down the RNA, DNA, etc. And then you have the phospholipases. One of the important phospholipases is what? The phospholipase A2. Okay, and what causes the activity of that? Okay, it is the trypsin that causes the conversion of prophospholipase to phospholipase A2. Okay, so what does they do? They break down the phospholipids, okay, and breaks down into the individual free fatty acids and lysophospholipids. Okay, so that is the major actions of these pancreatic enzymes. So remember what do they do and what helps to cause the activators. So important to remember about the activator like enteropeptidase, trypsin cause all the remaining activation. Okay, so let's move forward and see what is the how is this secretion happening. Okay, so secretion of bicarbonate by the pancreatic duct. Okay, the bicarbonate, how the pancreatic duct is causing that. Same like the reaction that we have seen in the parietal cell. What happens? It takes the carbon dioxide, right, and makes it to react with water. You have the carbonic anhydrase, and the carbonic anhydrase will cause the reaction between the carbon dioxide and the water and forms what? Carbonic acid. Okay, so this carbonic acid immediately get dissociated into bicarbonate and H plus. Okay, so the bicarbonate is actually transferred into the lumen of the duct and then it is being secreted into the lumen of the intestine. Okay, but what happens to the hydrogen ion? Hydrogen also is like secondary active transport takes place for the hydrogen ion, and the hydrogen ion is secreted to the blood. So hydrogen ion goes to the blood. Okay, so what does it do in the blood? It causes the neutralization of the blood that was coming from the stomach. Okay, it was coming from the stomach, and this hydrogen neutralizes that blood that is coming from the stomach, which is called the postprandial alkaline tide has happened. So alkalinity is neutralized by this H plus. Okay, so that's the importance of the bicarbonate secretion from the pancreatic duct. Okay, now let us see the regulation of the pancreatic secretion. Okay, how we regulate them. Regulation of the pancreatic secretion, which includes mainly the hormones and the neurotransmitters. Okay, so very less, like neuronal is very minimal. Okay, so regulation of pancreatic, especially the exocrine pancreatic secretion, which includes mainly three things, that is the acetylcholine, CCK, that is the cholecystokinin, and the secretin. Okay, acetylcholine is coming from the parasympathetic wave. Okay, that is the vagus nerve, the 10th cranial nerve. Okay, the 10th cranial nerve or the vagus nerve, and the other cholinergic enteric nerves. Okay, and causes the increased secretion of the enzymes, mainly the enzymatic part. Same way, the CCK also secreted from the eye cells causes the SNR cells to produce enzymes. Okay, so both acetylcholine and CCK helps to increase the production of enzymes, whereas the secretin causes the coming from the S cells cause the secretion of what? Bicarbonate. Okay, because it stimulates the duct cells and secrete large volume of solution with a high bicarbonate. So secretin. So maybe like just when logically if you think, because the secretin is stimulated by the acid. Okay, the acid is stimulating, so the acid has to stimulate what? The bicarbonate. You want to neutralize it with the help of bicarbonate, whereas CCK is stimulated by what? The fat and the other protein digested or the carbohydrate digested particle. The fat is causing the stimulation of cholecystokinin. Fat is stimulating the eye cells to secrete the CCK. So what does the CCK should do? Increase the enzyme secretion that will help to digest the fat, help to digest the proteins and the carbohydrate. Okay, so that is very logical thinking. Okay, Now coming to acetylcholine. Okay, acetylcholine is coming from the parasympathetic. So it's like more like a preparatory effect that is coming from the vagus. Okay, so let's see what are those like preparatory effect. How do we call? We call it as like three phases. Cephalic phase. Cephalic phase is the one that is mainly controlled by the cephalic phase is mainly controlled by the vagus nerve, okay, and by the acetylcholine. So phases of secretion include cephalic phase, then the gastric phase, then the intestinal phase. Okay, so so the vagal stimulation, okay, so that is first the cephalic phase, the vagal stimulation, okay, that is releasing enzymes, okay, and acid from the stomach releases secretin, okay, from the wall of the duodenum, and then fats and amino acids cause the release of CCK. Okay, so acid is causing the secretin, fats and amino acids cause the secretion of CCK. Okay, and the secretin, and what do they do? The secretin and the CCK, okay, absorbed into the bloodstream because they are hormones. They are not like directly coming over there. They are absorbed into the bloodstream. Okay, because they are secreted as hormones, not coming into the lumen. Okay, then what do they do? The secretin causes the copious secretion of pancreatic fluid that is rich in bicarbonate, whereas CCK causes the secretion of enzymes. Okay, so remember these three things: the role of acetylcholine, the role of the secretin, and CCK. Acetylcholine coming from the parasympathetic. Okay, so that's the importance of the regulation of the pancreas. Okay, so now we have finished with the pancreas. Okay, so now let us move to the liver. Okay, so we have seen with the pancreas. Okay, so the liver. Okay, so in addition to the digestive function and the production of the bile, the liver have like many other functions. Okay, so let's discuss a little bit about the other functions of the liver, but mainly we will concentrate on formation and secretion of the bile. Okay, which is about like 500 ml secreted per day. Okay, and what are their role? That is the most important thing for the digestive system. The other roles which you study, mainly the nutrient and the vitamin metabolisms, okay, especially in the biochemistry, you study about the glucose and other sugars. So as soon as the glucose or the glucose is absorbed, what happens? It goes to the liver, and the liver performs the other metabolic activities. What to do with the glucose, whether to store it or like all other activities, mainly determined by the liver. Okay, so that circulation that goes directly from the intestine to the liver is called as what? Enterohepatic portal circulation. Okay, it's a portal circulation. Okay, and brings all these nutrients. Okay, amino acid, lipids, but the lipids mainly also reach the liver, and the lipid metabolism is also controlled within the liver. But as far as the lipid absorption is concerned, it does not get absorbed into the enterohepatic circulation. Instead, it goes into the lymphatic circulation. We'll discuss about the lipid absorption slightly later. The cholesterol, lipoprotein, fats, soluble vitamins, what is all of them under the liver. Okay. Another important function of the liver is what? Inactivation of the various substances like toxins, steroids, and other hormones are inactivated. Okay, for the elimination. Then synthesis of plasma proteins, except the globins. Immunoglobulins is not synthesized from the liver, except the immunoglobulin. All other plasma proteins are synthesized from the liver, which includes the acute phase proteins, albumin, then the various clotting factors, and the steroid binding and the hormone binding, other hormone binding proteins. Okay, are all coming from the plasma protein that is coming from the liver. Okay, and for the immunity, the liver contains a specific cell called as the Kupffer cells. Okay, so but these functions are not the major concentration of our lecture. So we are trying to go and look into the details of formation and secretion and the functions of this bile. Okay, as far as the digestive system is concerned. Okay, so let's study about the bile secretion, liver and the bile secretion. Okay, so liver secretes the bile. Okay, what are the things that is present in the bile? Okay, bile contains bicarbonate. Okay, that is coming especially from the ducts. Okay, what is the role of this bicarbonate? Again, same like from the pancreas, you have the pancreas also secretes a lot of bicarbonate. Same like that, here also you have bicarbonate to neutralize the acids that is coming from the stomach. Okay, so the most important role of the bile, the content of the bile is the bile salts. Okay, bile salts are a combination of the bile acids. Okay, bile acids conjugated with the other substances like glycine, other amino acid. These are the amino acid glycine or taurine, and also conjugated to sodium or potassium and made it into salts. Okay, of these acids are secreted in the bile. Okay, so we will discuss about the details of what is cholic acid and chenodeoxycholic acid slightly later. Okay, and what is the role of these bile salts? They helps in emulsification. Okay, emulsification of the fat. Emulsification means it's not breakdown of the fat. Okay, breakdown of the fat, large molecules of the fat, like large droplets of the fat, will be broken down by the segmentation contraction and the peristaltic contraction that takes place in the intestine. But when they break down, for example, when you have a large droplet of fat, when they break it down into smaller particles, they will have what? Surface tensions, attraction between them. They attract to each other. Okay, they attract to each other. Okay, so they will again tries to come back and coalesce and become the large droplet again. Okay, so when it stop the one, so that is why when you have a large droplet, the surface area is decreased. So in order to have a large surface area, you need them to be remain separated. So what they happen? The small droplets of the fat will get covered by what? The bile salts. Okay, and they prevent the rejoining of them. Okay, they don't allow them to come back and coalesce. Okay, instead they keep them separated. So that is what is called as the emulsification. Okay. And then also after the emulsification, it helps the pancreatic lipase and the colipase to act and cause the digestion of the lipid over there. And then after that, it also helps for the absorption of the fatty acids, monoglycerides, cholesterol and other lipids by doing what? By the micelle formation. So what is the role of these micelles? These micelles carry these digested lipids from the center of the lumen to the brush border. Okay, so that carrying, so we sometimes refer that as the ferrying action. So they ferry the digested lipids from the center of the lumen to the brush border. Okay, so in addition to these like bile salts, also for the emulsification and the micelle formation function, we have other substances, which includes the lecithin and the cholesterol, also the bile pigments. You will study like more about the bile pigments in your biochemistry, which are the bilirubin and the biliverdin. This comes from the breakdown of the heme molecule. Okay, and this is the one that is responsible to give the yellow color, the bile pigments. Okay, like in the urine, in the fecal matter, you have the yellow color that is coming from the bile pigment. Okay, so that comes from the breakdown of the heme molecule. Okay, so and also when you get like disease conditions like jaundice, the icterus, that yellowishness, especially seen in the eyes. Okay, so those are coming from the bile pigments. Okay, but mainly how we break down the heme metabolism, heme catabolism, that you will study in the biochemistry. Okay, but it's important point, because like for example, when a physiology question comes, ask you there is lack of bile, so what will happen to the fecal matter color? If the bile is not coming into the intestine, they will not have the yellow color, right? Okay, so that the stool or the fecal matter will be clay colored. Okay, so that point you have to remember, that is the importance of the bile pigment. So this is like a waste product that is coming from the catabolism of the heme, and it is for the elimination that is coming into the bile. Okay, not like a functional part. The most important functional part is the bicarbonate for the neutralization, and then you have the bile salts. Okay, and the lecithin also helps for the emulsification. Okay, so these bile salts and lecithin helps for the emulsification and for the micelle formation. Okay, so let's discuss a little bit more. Okay, about the content over here. Okay, so composition in the hepatic duct. Okay, the hepatic duct bile is slightly different from that of the bile from the gallbladder. Okay, so from the hepatic duct it actually goes to the gallbladder, and what happens in the gallbladder we will see. Okay, so here mainly you have very high level of water. Okay, the water is very high when it is coming from the bicarbonate. Okay, so the bile salts, okay, bile pigments are there, cholesterol is there, other inorganic salts, okay, lecithin importantly, okay, alkaline phosphatase, all these are there. Now, so the main thing over here you have to remember is that when it is coming from the hepatic bile, the duct bile, it has a very high quantity of water. Okay, so we will discuss what happens in the gallbladder. Okay, we will discuss what happens in the gallbladder. So let us discuss about this bile acids. Okay, so what are the various bile acids? So we have already told the name of the bile acids, okay, that are what the cholic acid and chenodeoxycholic acid. Okay, so when you conjugate, how the name will be, if you conjugate to the glycine, we call it as glycocholate. Okay, so and also it is associated with sodium or potassium, so it can be called as sodium glycocholate as an example, or potassium chenodeoxycholate. Okay, so this is how we name the bile acids. So we have two different types of bile acids. One is the primary and the other one is secondary. Okay, so what is the major differences within the primary and the secondary? Primary is produced by the hepatocytes. Okay, whereas when the primary goes to the intestine, what cause the bacteria cause the conversion of the primary to secondary. The bacteria in the intestines can cause the conversion of the primary to secondary. Okay, so from the cholic acid, what is formed? The deoxycholic acid is formed by the bacteria, and chenodeoxycholic acid is converted to what? The lithocholic acid. Okay, so how do we do that? Okay, so normally when we secrete this cholic acid, you can see the hydroxyl group. This is already present in the cholesterol. So that means the bile acids are produced from what? From the cholesterol. Okay, this is synthesized from cholesterol. The bile acids are synthesized from hydroxyl group that is present on the third position is already present from the cholesterol. Okay, but during the process of the synthesis of the conversion of the cholesterol to bile acid like cholic acid, what the liver do, they add a additional hydroxyl group to the 7th position, add another hydroxyl group in the 12th position, and add a carboxyl group to the end. Okay, so what happens over here, all these like hydroxyl group or the carboxyl groups, okay, all these are water loving. Okay, whereas this ring structure, this ring that is present over here, all this ring that is present over here, the 4 rings, okay, they are the fat loving part. Okay, the ring is called as a cyclopentanoperhydrophenanthrene ring, that is the fat loving part, but the hydroxyl groups are water loving. So that is the important property of this bile acid. They have water loving part and the fat loving part. So that water loving part and the fat loving part helps to emulsify. Okay, the emulsification. So that means the water loving part will be towards the water and the fat loving part will be towards the fat and helps to become miscible in the water. Okay, so that is the importance. Okay, so as much as like possible, if you have more number of hydroxyl group or the carboxyl group, that is very good. And also in addition to the hydroxyl or the carboxyl group, the glycine or the taurine and the sodium and the potassium, they are all like water loving part. Okay, so now what we have, that is why it is important to convert the bile acid into the conjugated bile salt. Okay, and what happens when you have a lot of carboxyl group on the 3rd position, 7th position and the 12th position, like the cholic acid, what you have, that is the very high percentage. Okay, because this is very useful. So almost 50% of the bile will have which bile acid? The cholic acid. Okay, and little bit also is produced from the liver, that is the chenodeoxycholic acid, which does not have a hydroxyl group in the 12th position. Okay, so now what does the bacteria do? The bacteria will cause the conversion of cholic acid to deoxycholic acid by removing the hydroxyl group from the 7th position. So this does not have an hydroxyl group in the 7th position, but the 3rd position and the 12th position that remains. So it has still have the 2 hydroxyl group. Okay, so still can be useful. Okay, so it is almost like 15%. But when you convert the chenodeoxycholic acid into lithocholic acid, what happens? The hydroxyl group from the 7th position is removed by the bacteria, and already the 12th position there is no hydroxyl group already. Okay, so it will have only the hydroxyl group in the 3rd position. So it is useful because it's very less hydroxyl group. So that is why the percentage of that will be very less in the bile. Okay, so now importantly, see, the primary is produced from the hepatocytes, but the secondary is produced by the bacteria after it has gone to the intestine. So what do you expect? Do you expect the hepatocytes, when they secrete the bile, will it contain the secondary or will it contain only the primary? If you think that if it contains only the primary, no, it's not like that, because the secondary will be also seen in the secreted bile. Okay, because what happens, the secondary bile is coming back to the liver from the intestine by the enterohepatic circulation. Okay, and when it comes back to the liver, what happens? The liver again secretes the secondary. Okay, secondary is there secreted but very little. Okay, what also happens, the secondary, when it comes back to the liver will convert the secondary back to the primary also. Okay, so that is the role of the liver. Okay, so now let us see the importance of the role of this enterohepatic circulation. Okay, enterohepatic circulation. Okay, so how do we absorb this bile? Okay, this bile, the enterohepatic circulation of the bile. Okay, so which part it is getting absorbed? It is getting absorbed from the ileum. So it is getting secreted in the duodenum. This is the duodenum. Okay, it's getting secreted in the duodenum, and from the duodenum, what happens? It helps for the emulsification, micelle formation, digestion, and absorption of fat. And by the time it reaches to the ileum, what happens? It is taken back by what? The sodium bile salt co-transporter. Okay, which is a secondary active transport. So you need the primary active transport, that is the sodium potassium pump on the basolateral membrane. Okay, so the sodium bile salt co-transporter system works, and then they reabsorb the bile salts. Okay, and then it is taken back. So what happens over here? You can need to synthesize only like 0.2 grams per day additionally. Only few that has been left into the large intestine. Only 0.2 gram. But on a day, how much is being secreted? 3.5 gram is being secreted, but from that only like 0.2 is like getting eliminated. What this remaining is only need to be synthesized. Okay, so if a person have like a very high cholesterol, okay, one of the important ways to eliminate the cholesterol is as bile salts. Okay, to convert the cholesterol into bile acid and get eliminated. Okay, if you want to stop this absorption, either blocking the sodium bile transport or help the bile acids to attach to some fiber and get eliminated. So one of the important advice given to a patient who have increased cholesterol, hypercholesterolemia, is to consume a lot of fiber. So when the person is consuming a lot of fiber, what happens? That fiber will combine with the bile acid and get eliminated. Okay, and then what happens? The cholesterol will be more number of cholesterol is required for the synthesis of this much. Okay, instead of 0.2 gram per day, that synthesis of the bile acids will be increased. So there will be more utilization of the cholesterol. Okay, so that is one of the important applied aspect of this enterohepatic circulation. There are also drugs, not only just the fiber, like cholesterolamine, so that like a active particles that can bind to the bile acid and get eliminated. Okay, so that the cholesterol utilization will be increased, so that the blood cholesterol level can be decreased. Okay, so let's see the role of this bile acids. Okay, so how do they form? Okay, so this is the structure like a micelle formation. Okay, this is a cross section of the micelle. Okay, so which is helping to transport. Okay, so you can see the bile acids are there on the, it's like a discoid, like a cross section, like a coin shaped structure. Okay, and this hydroxyl group you can see on the periphery, the peptide bonds, the glycine and the taurine bonds, and the carboxyl group. Okay, all these are the water loving. Okay, this is like the water, and they are coming on the periphery. Okay, and also you can see the phospholipids. Okay, the phospholipids contains what? The phosphate groups. Okay, the glycerol and the phosphate group, these are all the water loving part, but these chains that you see downwards, they are the fat. Okay, and the free fat, the digested free fatty acid, the digested cholesterol, etc., will be there on the center. So this is getting transported. Okay, so this is getting transported, and when this micelle reaches the brush border, what will happen? The brush border will break it out and absorb all the free fatty acid and the cholesterol. Okay, and then the micelle will again come back, and which will get filled with the digested fatty acids and the cholesterol. So this is keep shuttling. Okay, so in the intestine. Okay, so now let us discuss the differences between the hepatic bile and the gallbladder bile. Okay, so what is the role of this gallbladder? Gallbladder stores the bile. Okay, gallbladder helps to store the bile. So the bile production is a continuous activity from the liver, but the sphincter of Oddi is not open. Okay, so the bile cannot always keep going into the intestine, so it has to be stored somewhere. So that happens in the gallbladder. Okay, so the bile is secreted by the hepatocytes in the bile duct, which drains into the duodenum. But between the meals, the duodenum orifice of this duct is closed, and the bile flows into the gallbladder. Okay, so bile flows in the gallbladder when the sphincter of Oddi is closed. Okay, and where it is stored and concentrated. So previously we have seen there is 97% water, but whereas the gallbladder, what does it do? It does not have like the urinary bladder, does not have the epithelium or does not have the muscular nature. to in order to expand. Okay, so that property is there for the urinary bladder, but for the gallbladder it cannot expand in nature. It does not expand like that. Instead, what they do, they keep absorbing the water. They keep absorbing the water and make it more and more concentrated. Okay, so that is why the gallbladder bile have only 89% of the water. So when the food enters the mouth, the sphincter of around the orifice relaxes. There is the sphincter of Oddi relaxes, and when the gastric contents enter the duodenum, the hormone CCK, okay, the CCK from the duodenum, CCK causes the gallbladder to contract and cause the secretion of the gallbladder bile. So you can see over here the hepatic bile. Okay, the hepatic bile percentage of the solids is only 2 to 4, whereas when it reaches the gallbladder it becomes 10 to 12% because the water is being absorbed. Okay, so the bile salts, okay, that is the same thing. Okay, 10 to 20 only, but here it has become almost 10 times more. Okay, so that is like 5 to 10 times more, that is like 200 millimoles per liter. It is increased because the water is getting absorbed. Same way for the pH also. But over here what happens, when the bile duct is being secreted, you have a lot of bicarbonate, so it will be more alkaline, whereas it will be more neutral or acidic pH when it is coming from the gallbladder. So there will be less bicarbonate, so because the bicarbonate is coming from the duct, okay, not from the gallbladder. Gallbladder does not do any secretion, it only causes absorption. Okay, so let's discuss about how this is being regulated. Okay, how the secretion from the liver is regulated. Okay, so secretin via the bloodstream stimulates the liver duct secretion. Okay, and that means secretin is coming from what? The acids. Okay, so the acid stimulates, so you need a lot of bicarbonate, and bicarbonate is secreted from the duct. That is why the secretin is stimulating the duct. Okay, so the another thing is like bile acids via the blood stimulate the parenchymal secretion. Okay, so the bile acid, when it comes back through the enterohepatic circulation, bile acid itself act as a regulator. Okay, it itself stimulates its own production. Okay, and the vagal stimulation causes the weak contraction of the gallbladder. Vagal stimulation through the acetylcholine. So but it is there, but it's also a stimulus, little bit weaker compared to the CCK. Okay, CCK is the major regulator for the gallbladder. Okay, via the bloodstream coming causes what? The gallbladder contraction and the relaxation of the sphincter of Oddi. Okay, so that the gallbladder bile is going into the in duodenum. Okay, so what is done? What is the role of the gallbladder? The bile is stored and concentrated up to 15 times in the gallbladder. So that is the role of the gallbladder. Okay, so regulation of the biliary secretion. Okay, the regulation of biliary secretion. We have to know about two terms, that is called as the cholagogue and cholerectic. Okay, cholagogues means those substances that relax the sphincter of Oddi and also cause the contraction of the gallbladder. The gallbladder contraction and the relax the sphincter of Oddi. And the most important cholagogue is what? The CCK. Okay, the CCK that comes from the duodenum. Okay, by the stimulation of the fatty acids and the amino acid that is in the duodenum, you get the secretion of CCK. CCK then cause the contraction of gallbladder as well as cause the relaxation of the sphincter of Oddi, and they are called as the cholagogue. And what is the meaning of the word cholerectic? Okay, the cholerectic means that cause the increased bicarbonate and the increased production of the bile from the hepatocytes. Okay, the duct activity, so it increases the bicarbonate content of the bile. Okay, so and which are the major cholerectic? The vagus nerve and the secretin. Secretin is by the acid, so you can imagine you need a lot of bicarbonate. Another important physiological is the bile salts. Okay, bile salts act as an important physiological corrective. They come through the enterohepatic circulation and stimulate it. Okay, so those are the major important points of the biliary secretion. Also remember, when there is a question that is asking you about a gallbladder and the gallstone. Okay, so what can cause the contraction of the gallbladder? There is CCK. Okay, so for example, if a person is having a gallstone, what will happen? The CCK will go and cause the contraction. So if the person eats fatty food, If the person have a stone and then eats the fatty food, what will happen? There will be more CCK and cause more contraction of the gallbladder. So when there is more contraction of the gallbladder, the pain will be enhanced. So remember that point, okay? For the exam point of view, that question is very important. Okay. So now what we have discussed is about the small intestine and its secretions, its secretions not only from the small intestine, also the secretions from the outside, like from the pancreas, and also discuss the role of the liver. Okay, so and also we have seen a little bit movements and the regulations of these secretions. Okay, so next what we in the next video what we will discuss is about the digestion and absorptive activities that takes place in the small intestine. Okay, thank you.